Today we're going to talk about the first two steps in cell respiration. So first of all, there's going to be three main steps. So let's do a quick walkthrough of all three, like an overview. So the first step is called glycolysis. This is the only step that happens in the cytoplasm, meaning you do not have to have mitochondria for this step. It also doesn't require oxygen. So even bacteria, which do not have mitochondria, can do this step of glycolysis. So it starts by breaking down glucose into two molecules of something called pyruvate. Now, pyruvate is 3 carbons and glucose is 6. So if you think about glucose, 1, 2, 3, 4, 5, 6 carbons. I'm not drawing, it's actually, remember, glucose is C6H12O6. I'm just drawing the carbons here. It's got a bunch of steps, but ultimately, it just splits glucose in half. And that's what glycolysis means. It means to split glucose. So, and these 3 carbon compounds are going to be called pyruvate. The second step is really 2 steps. There's a little mini step called the oxidation of pyruvate, and that happens as these pyruvates go into the mitochondria. And then there's a step called the citric acid cycle or the Krebs cycle. And by the end of the citric acid cycle, all of the glucose, C6H12O6, has been completely broken down into carbon dioxide. So the carbon dioxide is done. But the hydrogens from the glucose are actually trapped in these coenzymes that we're going to talk about, and those are going to go to the very last step. So the only thing left by the end of step 2 is going to be the hydrogens from the glucose. The carbon and the oxygen are gone. And then in our last step, which is called oxidative phosphorylation, it's the ETC or the electron transport chain. It involves something called chemiosmosis. We'll get into that, but not today. These electron carriers, NADH and FADH2, see the H's here? These are all the hydrogens from the glucose. These are going to basically go through a reaction in the mitochondria that's going to generate a majority of the ATP for cell respiration. So, and it even says that here. Most of the ATP made by cell respiration is going to be made in this third step by this thing called oxidative phosphorylation. At the very, very end, the hydrogens that we're starting off in the glucose are going to end up in water. So our overall formula, remember, is C6H12O6 plus 6 oxygens, and it's going to make 6 carbon dioxides and 6 waters. And it turns out, this is actually a net reaction, so there's a little more to it than this, but ultimately the carbon and the oxygen from the carbon from glucose will become carbon dioxide, and the hydrogen will hook up with this oxygen and form water. So this is sort of a picture. So again, glycolysis, glucose will get turned into this 3 carbon molecule called pyruvate. That's going to happen in the cytoplasm. And then the oxidation of pyruvate and the citric acid or Krebs cycle are going to happen in the mitochondria. And then our last step, ETC or oxidative phosphorylation, will also happen in the mitochondria. So glycolysis is the only step that does not require the mitochondria. Okay, so let's jump into this. All right, so glucose. Glycolysis means to break glucose. Glyco means glucose, and lysis means to break. And so this happens in the cytoplasm, and what's going to happen is 1 6-carbon glucose is going to be split into 2 of these molecules. This diagram is pyruvate. You do not have to be able to draw that, so don't worry. It's an 8-step process. So this is not, even though ultimately glucose just gets broken in half, it's not a single step. It actually happens in 8 steps. Now it requires 2 ATP to get it started. So the first few steps actually require energy, but then in the end it makes 4. A lot of times on a test you're asked how many net ATP, meaning after you take away what it requires to run, how much ATP are you actually ahead at the end? You know, what's your profit, so to speak? Only 2 ATP. Because since it costs 2 to start it, even though you make 4, you're only ahead by 2. Now, it also makes this coenzyme I mentioned. This is called NAD, It stands for nicotinamide adenine dinucleotide, but it's a coenzyme, meaning it helps enzymes. And all you really need to know is that when it picks up this hydrogen, it's going to be able to carry this to another location to later on generate more ATP. So in the beginning of glycolysis, it looks like this, NAD+, and then it picks up hydrogen and becomes NADH. This is very quick, and it also does not require any oxygen, which is important. That means even if you were oxygen deprived, you could still run this process of glycolysis, but it wouldn't keep you alive very long for a couple of reasons, but one of them being you don't make very many ATP. You only are going to make about 2 ATP per glucose, and that's not enough to keep you alive for very long. This is sort of an overview. Again, glucose in the cytoplasm or cytosol gets broken into 2 pyruvates. It makes 2 NADHs from NAD. Remember, this was NAD+, and it becomes NADH. These hydrogens are coming from the glucose. And then it also makes a net of 2 ATP. Technically, it makes 4, but it costs 2. And here's sort of the summary. So 1 glucose goes in, 2 NAD+, this is this coenzyme without the hydrogen, and 2 ATP are necessary to get it started. It makes 2 pyruvates. It makes 2 NADHs. These H's are coming from the glucose. And it makes 4 ATP, but remember, your net gain is only 2 ATP because it cost you 2 to get the process started. And this is showing it again, a different picture, showing multiple steps, ultimately 2 pyruvates. Sometimes pyruvate is also called pyruvic acid. And here they're showing the NAD plus getting turned into NADH. Again, these H's are coming from the glucose. This is showing 2 ATP going in and getting broken, but 4 ATP being made. And this diagram also shows, again, the same thing. The first few steps require energy. Here's the 2 ATP that go in. But then 4 ATP come out. And I doubled this, but I didn't realize it already had doubled all these numbers. So it's not actually 2. It's, in other words, they originally had a single picture, and I doubled the picture because I wanted you to see that it's each pyruvate is going through this, but I didn't realize they had already doubled these numbers. So this is actually, these should all just be 1, 1, 1, 1. Everything should be a 1 here. But then when you add them all up, 1, 2, 3, you would make 4 ATPs. That's what I was attempting to show. It's a bad drawing. Okay. Now, at this point, there's a choice. And it really depends on whether there's oxygen present or not. So if there is oxygen, if there's not oxygen, then what's going to happen next is called anaerobic respiration without AN in front of. Aerobic means with oxygen, so AN in the front means without oxygen. This is also called fermentation. We're going to talk about this later. We're going to jump into what if oxygen is present next. So if oxygen is present, we're going to continue with our cell respiration. It's going to be called aerobic because it's going to use oxygen. Think about it using air, even though that's not the translation. All right, so our first step here is that the pyruvate from the glycolysis is going to enter the mitochondria. So this is a mitochondrion. It has 2 membranes, an outer membrane and an inner membrane. These folds are called cristae. They're going to be important. And this center is called the matrix. So 2 membranes, folds called cristae, and a center part called the matrix, which looks like it's full of water, but it is full of solution. It's not really pure water. All right, so this step in between. So pyruvate, this is the pyruvate. Remember, this is a 3-carbon molecule, 1, 2, 3 carbons. As it enters the mitochondria, it's going to get turned into something called acetyl coenzyme A. You don't really have to understand the details of it. It is going to lose a carbon dioxide, and it's also going to make another NADH. So it says here 2 carbon dioxides are released and 2 NADHs are made. The reason that it's 2 is because you had 2 pyruvates. So in other words, you have 1 pyruvate, you have 2 pyruvates, right? Because glycolysis made 2 of these. So for each one, you're going to make another NADH, and you're also going to make a carbon dioxide. So that's this little step. It's called oxidation of pyruvate because the pyruvate, it's called being oxidized because it's losing, well, we're not going to get into oxidation and reduction, but it's called oxidation of pyruvate. All right. And then our next step is the Krebs cycle. So it's also called the citric acid cycle because that's actually the first thing that's made in the Krebs cycle is citric acid. So two names, Krebs or citric acid, it's the same thing. And it happens in the matrix, which remember is this center area of the mitochondria here. This is the matrix. So it's going to take the acetyl-CoA, this was what was made in the previous step, and it's going to convert the remainder of that to carbon dioxide. It's going to make some more NADHs, and it's going to make another molecule called FADH2, flavin adenine dinucleotide, and it's going to make 2 ATP. So at this point, all of the glucose has been turned into carbon dioxide, and what's left is NADH and FADH2. That's what's going to go to our very last step. Is going to be these two coenzymes with all these hydrogens from the glucose. All right. So to summarize, this is showing the Krebs cycle in the mitochondria, or the citric acid cycle. This is the oxidation of pyruvate step. And then they're showing, you're not going to be responsible for knowing the numbers, but it makes a bunch of NADHs. All of these hydrogens are going to be important in the very last step. This is showing the same thing. This is the cytoplasm. So you had the glycolysis, then the oxidation of pyruvate is this step as it enters the mitochondria. And all of this here, this is the Krebs cycle. It's a whole bunch of steps that you don't have to know. So this is the oxidation of pyruvate. So remember, glucose had 6 carbons. Pyruvate has 3. So this is the pyruvate from glycolysis. A carbon dioxide breaks off, so there goes a carbon from it. You make NADH, that's some of the hydrogen from it. It hooks up with this coenzyme and it makes acetyl-CoA. You end up with 2 of these because you had 2 pyruvates, because glucose split into 2 pyruvates. And then each of those goes into the citric acid cycle or Krebs cycle, makes some more carbon dioxides, some more NADHs. Again, from NAD, it's picking up hydrogens from the original glucose, this FADH2 as well, and you do make a couple of ATP. So this is the whole cycle. All right. So our last step is going to be the electron transport chain. So we'll talk about that in our last lecture.